All right, for this assignment, after you've watched the video on actually how to do this, you need to go find the files for this assignment where you're going to annotate the lacAnnot. What you need to do is you need to go find the file that is labeled EF1368884.1. Now what this is the E. coli K12 lacAnnot. Well sequenced. This is the standard one that's used in research. If you order this, this is by far the most common lacAnnot and E. coli. E. coli K12 is the one that you use in labs everywhere. It doesn't make you sick if you get it in your food or anything. It's got a genome that's been kind of engineered to work easily with molecular biology tools. So this would be what you would use in a microbiology lab. So you need to go get the E. coli K12 lacAnnot, and you need to get something called the GenBank file instead of the FASTA. So when you download it, and normally you download the FASTA, instead you just choose the GenBank file. If you want the FASTA, which you're going to need for this this assignment, you would click here, and of course it takes you to the FASTA. So your assignment is, of course, to annotate the FASTA. So, for instance, if I take the FASTA, if I go get the FASTA, and let's download it. I'll get the number with it and create the file. And I'm going to go in here and grab it real quick. But normally I tell you not to open these with Word or whatever. But in this particular case I am, because we want to highlight certain things. And if you watched the other video, you realize that this is a strange sequence. Normally the start codon is AUG, and this one doesn't have an AUG as the start codon. It does code for methionine, but the sequence, sometimes the ribosome will attach to things that we don't understand. It attaches right here, and I'm going to change the color of these. It's GTG here, and I show this in a previous exercise, in a previous video. And we also decide in that video that probably the Shine-Delgarno, or the Shine-Delgarno is here. So I'm going to change that to a different color, blue. And this is kind of what you should be doing. Let's do this so that it stands out more. Let's make all of these blue, and let's make the background yellow. All right, that'll stand out. And then for Shine-Delgarnos, and here's our start. So let's make this, what stands out? Let's make the background blue here. All right, something like that. So there's our start codon, red with a blue background, our Shine-Delgarno, yellow with a blue background. And then we will go, and we decided in that other video that this was our Tata box. And so a Tata box, maybe we make it orange with a, I don't know what goes with the orange here, with a green background. There we go. So here's our Tata, here's our Shine-Delgarno, here's our start codon. Our stop codon was further down, and here's where we get to use our GenBank file. Our GenBank file actually will tell us all of this. Let me get back to the GenBank file. Our GenBank file says, now this is the whole operon, and it does include I. And this is I right here, the lac repressor, that is I. And it starts at 70, and that's actually how I knew that this was the start codon. because these are 70 long. So 70 is right here, this last letter here on the line. And so since that's the start codon, we know that the rest of these, the protein coding region starts right there. Now let's go back here. Where does it end? So let's finish this off. That video didn't quite finish off everything for the I. It ends at 6,338. No, it doesn't. Sorry. That's the whole operon. It ends, this protein, just the I protein, ends at 1,161. So we have to go find 1,161. Well, these are 70 each. You'll have to count them up. But let's see. 10 of these would be 700. So here I'm just going to start right here. So 70, 140. So we're going to go 10 to get to 700. Well, say 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right, we go another one, and that's 770, 840, 910, 980, and then 1,090. I'm kind of lost. We're over 1,000 though. We should be getting close to the end. Let's see here. Where are we looking again? We're looking for 1,161. We should be close. All right, so you would look here and find the genetic code for G and Q. I don't know what that is. It's probably a glycine and I don't remember what Q is. You would find the potential DNA sequence. Remember, multiple codons though, so there are lots of them. There might be several here that could potentially code for this, and you would look for it down in this region. So we are, and did I lose my place? Nope, right here. So we're a little over 1,000. Somewhere in the next few lines here, you're going to find a sequence that could code for G and Q. So let's actually go do this. I need the genetic code from right here. I have it in this video. So let's see. Oh, but we don't have the letters. But G, pretty sure that's guanine. And it doesn't quite show here. I'm going to have to go get a genetic code real quick. Get an image. Go to images. Type in genetic code. And here we go. But we want the ones with the single letters. Let's see. Do we have this one? Where's one with the single letters? Yeah, right here. Okay. So we want G. So it can be GG something. Are there any other G's? Nope. So GG anything, followed by what's Q? Q is probably a rare one. Q is CA anything. So it would be GG anything, CA. The last one can be A or G. So it's GGNCA. GGNCA ending in an A or a G. So let's see if we can remember that. Okay. Maybe I'll do it this way so we can see it on the paper. Our decision was GGN, CA, and then an A or a G. And then what? An A? No, oh, already forgotten. An AG, no. Where was it? CA, then an A or a G. and then I do it like this, A or G. So this is what we're looking for, and we're looking for it, and of course I probably lost my place again. So 70, 2, 3, 4, 5, 6, 7, 8, 9, 10. Let's see if is it telling us, do I have something down here that tells me where I'm at? Nope. There are some word processors that do that, like Notepad++ does that. I would recommend that one if you haven't downloaded that one. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then we're going to go just a little bit further. So 770, 840, 910, 980, 1050, 1050, and then 70 more. Yeah, it's going to be right in here. Okay, so we are looking for a GGNCA. GGN, a GG, and a CA. Well, the way to do this is to go down here and do find, and let's look for our GGs on that line. And we do find all. Here's GGs, and then space CA. Here's a GGCA, but there's no N there. No. There it is. CANG. Yep, that one fits. So there is our GG. It's a GGG, CAG. And so we will highlight this one as our stop codon. So why don't we make that blue on red? Oh, I want to do it the other way. Red on blue. Okay. So start codon, stop codon. So this is your I protein. This is how I would expect you to do this. You'd create colors and tags like this, and then you would go and give me a legend, like what the colors are what. Like all the Tata boxes are green and orange, all of the Shine-Delgarnos are blue and yellow, red and blue, and here blue and red, back and forth, start and stop codons, that sort of thing. So to begin your assignment, I would like you to go and find all of the Shine-Delgarno sequences as the first assignment. I think, why don't you find Shine-Delgarnos and start and stops as your first assignment, and go through and do that. And here, so that one's an easy one, because you can do what? You can go use the GenBank file. The GenBank file will let you find the start and stops by counting the letters in this file. The GenBank goes along with this FASTA. So that's why it said it starts at 70 and it goes to whatever this number was, and that's how we did it. So you can go find Z, Y, and A in here, in this sequence. You find Z, Y, and A, and then look for right before them. All right, according to our rules that we have in the sheet, the other sheet, it's going to be within 30 base pair of this, and it's going to be an AG rich sequence. So you go find the start codons and the stop codons, and then right before the starts, look for an AG rich. These are called purines. A and G are purines. So a purine rich sequence right before, just like this one. And then we'll go to Tata boxes and things like that, and terminators and other sequences that we're interested in, like O and cap and all of those. We'll do those later, but let's first find where Z, Y, and A are. Here I give you where I is. We'll also need to find the terminator. This is the Tata box. There is a termination sequence in here. That's a little harder to find, and we'll do that one later.